thanks for the invitation to come and speak. I am at Imperial College, but I'm sort of moonlighting somewhat under a new initiative that is being set up at the Wellcome Trust Sanger Institute, the Centre for Genomic Pathogen Surveillance. This is an initiative between Imperial College and the Wellcome Trust Sanger Institute. And the overall aims of the centre are to provide data and tools for ongoing local, national and international use, for monitoring antibiotic resistance, undertaking of structured pathogen surveys, and ultimately to address questions of population and evolutionary biology. Now, I'm mostly interested in, currently, bacterial pathogens, and for most bacterial pathogens, many different strains or subparts of the population exist. And we need to be able to distinguish these and relate the distribution to their epidemiology. One classic example is antibiotic-resistant strains or clones of particular bacteria. This poses a number of public health challenges, which can be broadly categorised under three sections. Firstly, how do we identify high-risk clones? Secondly, how do we characterise these high-risk clones, for example, assess antibiotic resistance and virulence, transmissibility? And then how do we move towards management of high-risk clones? So I'm just going to detail one of the classic gold standard methods for strain characterisation of bacterial pathogens, Multilakas sequence typing. This has been around for a number of years and has become the gold standard nomenclature for describing high-risk clones of many bacterial species. This is a cartoon of a bacterial chromosome. And MLST assays variation at roughly 7 genes per bacterial species. And these genes have been chosen because they code for housekeeping functions. They're slowly evolving. They're crucial to the life of a species. So any sample that you take, these genes will be present. And the key method, collect a sample of bacteria, extract the DNA, PCR up these 7 genes, sequence the products, and then compare each of the sequences at each of the loci to an online database where we can assign a consecutive number or an allele number to each of the sequences of the 7 genes. And then each unique string of 7 integers can be uniquely defined as a sequence type. So this is an unambiguous identifier. So in this case, ST81 represents these allele numbers of these genes all the way back to the sequences. So it's a very shorthand. This is all digital data, so it can be stored in online databases and queried, etc. And these are the kind of data that we get. So there's a series of isolates. These are the allelic profiles that have been identified. And we can draw a very simple tree by comparing mismatches between allelic profiles. And this helps us identify not just single strains, but clones and closely related strains within a clonal complex. Now, these databases are available for many bacterial species. There are a number of websites that provide online resources, allele sequences, sequence types and epidemiological data for many bacterial species. And over the years, thousands and thousands of new STs and allele numbers have been assigned at these resources. And the traditional way of looking at these data are using trees, these mismatches, but when you start to get to very large online databases, trees become a fairly horrific way to actually look at a very large population. Once you get over a couple of hundred isolates, it becomes very difficult to actually interpret a tree. So new methods need to be developed to look at a bacterial population as a whole so that we can try and identify these clones in clonal complexes. One method, eBurst, was developed several years back. And actually, the way that bacteria evolve, this kind of method of drawing a tree, the bifurcating process of having an ancestor and then 2 and 2 and 2, is not a tremendously realistic way of depicting how bacteria evolve. So if we imagine in a population we have an emerging clone by MLST, it's identical. Over time it grows in frequency within a population. And then at some point one of those loci picks up either a point mutation or a combination and we get a new sequence type. Further over time we have different sequence type building up. And we can go from single variants, so these STs around the outside differ from the founding genotype at 1 out of these 7 MLST loci, or 2 out of these 7 MLST loci. And we can predict the founding genotype within a group as being the one that has the most single locus variants. And we can use these kind of methods, very simple methods, to divide up large populations of MLST data into more simple diagrams that try and predict a realistic founding genotype within a group. And we can inflict this algorithm on the whole MLST database. This is a population snapshot of the Staph aureus database, showing all the major and minor clonal complexes. And we can then use this to overlay information, for example, about presence or absence of drug resistance genes or epidemiological parameters. Now, eBurst has been used for a lot of different species, and actually you start to get an indication of what a population looks like by using this algorithm. So Staph aureus is a very clonal organism. Most of its evolution is through single point mutations. So it has these very nice demarcated clonal complexes. But when we look at other species, and this is largely dependent on the rate of mutation versus recombination, you get very different population snapshots. And this needs to be taken into consideration when we start looking at identifying high-risk clones. Burkholderia pseudomallei has huge amounts of recombination and lots of new allelic diversity. So bits and pieces are jumping around all the time. So you get one large straggly eBurst group. However, for Staph aureus, it's a bit simpler. And these eBurst groups have been used to define major epidemic clones. And this is a nomenclature that's widely used within the Staph aureus community. So people can talk about a particular sequence type, ST22, for example, which is epidemic MRSA 15, which is the major Staph aureus clone that's caused most MRSA across the UK and Europe and the rest of the world for many years. So most of the MLST databases have been built up by researchers or public health labs throwing new allelic diversity and sequence types into the database. However, one of the things that we need to move towards is more structured pathogen surveys. So this is a slide just summarising one approach. So pathogen surveys, we need a scalable structure, so multiple participants from multiple labs can send single or multiple samples using standardised sampling frameworks, ideally with a set of laboratories across a broad geographic area. And then we can use these data to assess population structure and identification of clones. So I'm going to go into one example of one of these large structured surveys using MRSA, Staph aureus. So Staph aureus exists in two major parts, methicillin-resistant Staph aureus or methicillin-susceptible Staph aureus. And this has arisen multiple times in the population. It's a directionality from top to bottom by the acquisition of, within an SSA, of a staphylococcal cassette which contains the gene that encodes for methicillin resistance. This is a paper from several years back which undertook a large structured survey across Europe. So between September 2006 and 2007, roughly 350 laboratories serving over 450 hospitals in 26 EU countries collected roughly 3,000 MSSA and MRSA. So each laboratory over a 6-month period collected the first 5 MRSAs and the first 5 MSSAs. And they were all genotyped using a method called SPAR typing. This isn't MLST, this is a single gene method. And this was chosen because it's cheap and very quick for national reference laboratories to utilise. So we wanted a way for all of the labs in the network to have access to that information and to be able to actually collectively interpret the results that were seen. So this is just a map showing the location of all the participating laboratories. So if we click on a location of a lab, this will then pull out the genotypes for the isolates that were submitted. So this is 4 isolates of a particular genotype, so this is again a SPAR type, and then we can view the distribution of that particular clone across the rest of Europe. So the green dots represent that for this clone, the isolates are methicillin susceptible, MSSA. Red represents that they're MRSA, and yellow means that at that location for this clone there's a mix of MRSA and MSSA. So this is a predominantly MSSA clone, very widespread across Europe. If we look at another clone, this is predominantly MRSA, and it's very geographically restricted, in this case around Spain. And when we looked at all the major MRSA and MSSA clones, these were the patterns that we saw. All the major MSSA clones are very widespread across Europe, and the MRSA clones are more geographically restricted. And this is indicative of a background susceptible population picking up the genetic element that confers resistance and then spreading locally between healthcare institutes. So this was an exemplar study just to indicate that using these kind of broad, widespread genomic methods, single gene in this case, we can potentially go all the way from understanding the population all the way down to potentially putting into place measures to stop the spread of a particular clone. But of course that was ages ago, and we're moving on to whole genome sequencing now. So the last 3 or 4 years have seen sequencing machines that allow us to potentially look at the entire genomic complement of an isolate. One of the most popular being Illumina, which produces millions of very short reads from a particular genome. And we can use these short reads to try and map to a reference genome. And then when we map the reads from a single isolate against a reference genome, we can look for single point mutations or differences. And this is the method that's very widely used currently within bacterial populations for assessing SNP differences between a subset of isolates. And again, in this case, trees are used to illustrate, just as an illustration really, to try and infer the most common recent ancestor. And this is used to investigate relationships over time and space. So most of the studies that have been undertaken, specifically within Staph aureus, have been looking at a single clone, so one clone that's been defined by MLST, collecting lots of samples of that clone, whole genome sequencing, and then seeing what extra resolution we get by looking at the larger sets of information. So back to this kind of eBurst snapshot of Staph aureus. So I mentioned ERMISA15, clonal complex 22 or ST22. So one of the first studies was undertaken by Matt Holden at the Sanger Institute. Got in contact with lots of laboratories and asked for samples of ST22 and then genome sequenced. So this is a tree based on a number of isolates from a number of different countries going back over a long period of time. And of course, if we know the date of isolation and we can work out the mutation rate by looking at the number of SNP changes, we can date specific genetic events when we look at the tree. So this is a multiply antibody-resistant clone, but when data were looked at, towards this particular point of the tree, there was an indication of a mutation in a particular gene that conferred resistance to fluoroquinolones. And then a very large set of isolates that contain that mutation and have spread very, very widespread. So this is another representation of the tree. These isolates here are the background population represented here, which don't contain resistance to fluoroquinolones. And this is a very large set, very quickly evolving, very rapidly distribution across a very wide geographic area. And of course, if we know that a particular gene has mutated or a particular gene's been picked up that confers resistance, we can actually then map the distribution. And when we map the distribution across the UK, spreading out, this, the origin of this particular burst here, when fluoroquinolone resistance crept into the population, coincided very nicely with the introduction of fluoroquinolones in Nottingham in the mid-1980s, and then a consequent spread, in this case across the UK, but then further across Europe. So from using whole genome sequencing, we have a much better resolution to try and understand the emergence and spread of this particular pandemic clone. And that's what it looks like using MLST. Brilliant. So this is the greater resolution that we get with whole genome sequencing that really allows us to delve further into the spread of a particular clone. This is another small study that was undertaken in a hospital. These were a set of isolates from an outbreak. These are a set of isolates from the rest of the hospital. The outbreak isolates cluster very closely together, indicative of transmission. And this is the standard way within a microlab of testing for antibiotic resistance. You have an agar plate, you spread your strain, you put antibiotic discs on, and then you see whether the antibiotic actually kills or the particular strain survives. And this table here is a standard antibiogram. So antibiotics at the top, particular strain, resistant or susceptible. But of course, if we have the whole genome sequence and we know potentially which genes confer for resistance, we can use that data to try and digitally predict antibiotic resistance. So this is at the bottom is the genomic resistance prediction compared to the phenotypic resistance prediction. So all of these studies take a huge amount of time, lots of postdocs collecting samples. And we need to move really towards a far quicker way of looking at very large sets of data. So we took, as an exemplar, a number of isolates from this structured survey that we did with Staph aureus to act as a representative population snapshot and a database to place new genomes into the context of a wider sample set. So we sequenced about 10%. This is the tree. We've covered most of the major clonal complexes. The little red boxes represent phenotypic, genotypic predictions, correlations, and then we have mobile genetic elements and dating of each of the subtrees. This is the clonal complex 22. But of course, this kind of figure's ridiculous because you can't actually tell a lot of detail from this particular tree. It would take an awfully long time to look through and read and delve down into interested claims. So we put together a very simple illustration tool that takes that data and then provides it in a way that the laboratories who have participated in the studies can actually look into the information. So this is again a very simple, lightweight web application. This loads a single file which contains the metadata and a Newick file that contains the representation of the tree. It's not really doing any calculation, but it's just a way of exploring the information. So again, each of these dots represents the location of a participating laboratory or hospital. This is the tree based on clustering SNPs within the sample set. This is a set of metadata at the bottom, so we can explore information about each of the samples. And this is just a set of buttons that allow us to overlay information on top. So if I change the colours to MRSA, again, the colour coding is that each of these positions, green represents that all of the genome sampled from there is susceptible, red is resistant, and yellow is where you have a mix of resistant and susceptible isolates. This is also updated on the trees so that we can see which genomes are resistant and susceptible. We can then overlay information such as the sequence type, the MLST type. So this is our EMRSA 15 MLST 22 clone. So if we look at that in a little bit more detail, we get this same kind of comet-like shape, which is susceptible background isolates and then a burst of resistance. Now this is a different set of genomes to that initial study, but we're seeing exactly the same phylogenetic shape. And if we delve a little bit further into this head of the comet, we can overlay information about the geographic origin. Now from Matt Holden's study, we know that EMRSA 15 or ST22 has arisen from the UK. And here we have a tree that shows two halves, UK, and then we have Portugal and Germany in here. And we can then infer potentially that we've had an introduction from the UK into Germany. In this case, if we look at the city, first location is Berlin, and then we see it radiating across Germany. Now this is really wholly based on the sample set. But this is a way of illustrating that we could be using these kind of methods to identify potential spread in as close to real time based on the sampling that we get. And actually this introduction into Berlin fits very nicely with anecdotal evidence from national reference laboratories in Germany for the origin of MRSA 22. Okay, so that data set was from a structured survey, population snapshot, static. That tool doesn't allow any new genomes to be added. So what we need to be doing is allowing or providing methods for new genomes to be placed within the context of very good background structured surveys. Ideally with prediction of resistance, virulence, determinants straight away, and potentially this kind of clustering method using trees to identify a little bit further. So we have a web application that we're launching quite soon. This is a demonstration version which contains whole genome data sets that have been published within Staph aureus currently. This is a set of Matt Holden's information, so the EMRSA 15 ST22. This is a very sort of generic interface. These are the genomes, these are the resistance profiles. Each genome has a report that gives similarity to other Staph aureus reference genomes. Shows you the sequence type, these are the MLST alleles, and then the predicted resistance profile based on genomic content. We can then view a tree of the clustering of this information. So here is the comet shape. Again, we can overlay extra information. We can highlight particular resistances based on certain antibiotics that have been predicted. And we can interact clearly between the tree and the map to delineate small subsections of the population. So how do we get our own information in? So this is a folder containing a number of genomes. So this is an assembled genome based on millions of short reads. There are assembly programs that are developed to try and string these reads together into a single contig or single chromosome. And then we have within the folder a simple CSV file that contains some metadata about these particular genomes, so the date of isolation, location, and source. So if I'm in a lab somewhere, I want to be able to firstly interpret my own information and then place it in the context of others. So if I drag these data across the web browser, this then passes each of those files, gives a small report on the overall content within each one. So this is for a single genome file, this particular one, the number of nucleotides, total number of contigs, etc. And this is a way of potentially assessing quality of the data that comes in. And the metadata is associated with that information. So this is all done in the browser before we submit this information for processing. So these data are then uploaded to a central processing server. And we have a number of modules that are run on each of the genome sequences. So firstly, we have a small module that predicts the MLST type, looks for the genes present, and then searches each of the alleles against a known database. We have a terrifically named module called parsnip, which is presence or absence of antibiotic resistance genes and SNPs. So this is a library of known resistant determinants that we can search against each genome. And we have two other modules that are used for drawing trees. This is a module that looks for genes that are present in every Staph aureus, so core genes. And then we have another one that looks for core and non-core genes, so genes that aren't only found in each one. So as each one finishes, we get one of these reports that I showed you, similarity to other genomes, the sequence types predicted, and then we have the antibiotic resistance profile. And as they're all finished, this is now, those 5 genomes have been processed. This is our digital antibiogram. It infers resistance for each one. Again, each one has a report. All of this information can be downloaded. The locations have been geocoded and placed on a map. And then we can also view a tree of the data that we've uploaded. So this is our collection of genomes and how they relate to each other. So this allows kind of local analysis of the information. And again, you have the capability of putting other information on top. Of course, we need to place this in context with other data, so we can merge our information with, in this case, a whole set of other ST239s that have been submitted from elsewhere. And this will do the calculations and merge this tree with a broader data set. And then we can investigate where our isolates fit on this particular tree. So I know where ours are because they're highlighted here. It's these two. But then we can look, for example, which are the closest isolates to the ones that we've just genotyped. And again, by clicking on the map, the tree, it delimits the data on the map, etc., back and forth. So we can see a broader global distribution of this particular clone. So there's lots of genomic data sets available currently, and one of our first initial aims is to try and make these data available in the same tools. How are we doing for time?
You have got 25 minutes, I think, or slightly less than 25.
Okay, perfect. I'll bore you with another tool that we're developing. So this is a tool we developed several years ago for mobile data gathering. This is a generic way of creating forms on a phone that can allow data to be geotagged and collected and centralized. So we initially built this for studies we were involved in, but we realized quite quickly that these kind of data gathering methods are fairly generic. So we have a website that allows anybody to come, build a project, design a form for data gathering, load that onto lots of mobile phones, collate the information with GPS and photographs, and then view data within a web application. So this is the website. We have two versions, one which allows single GPS and a single photograph per form, and then one that allows any number of GPS, photographs, videos, barcode scans, sound clips, etc. So if I just create a very simple project, we give it a name, RSTMH. This is now a permanent web page, project web page for our RSTMH project, and the first thing we need to do is create a data form, or a form for capturing textual information. So this is kind of what it would look like on Android or iPhone. These are the form types, the question types that can be answered. So if I drag across a simple text question, add something trivial. What is your name? An ID is essentially a column heading within a database, and then we can require that information. So if I then add a drop-down, this is a select one, whereby the question that you ask on the phone, you can select one of the choices. Favourite colour, for example. And then we just add our options. And then we want to see a pie chart of the information when lots of people gather data within our project. So we've, whoops, excuse me. Okay, so we've created a very simple 2-question project. Now I'm going to load this on the phone here, but these are the screenshots of what I'm actually doing. So the first thing you do is you load the project in. Oops. That's not going to work. One of the things isn't going to work, is it? So you load the project, and then first thing is to add a new entry. So each new entry is date and time stamped. You can add a GPS position, add a photograph, and then enter the form data. So this would be what the form would look like if it had loaded on the phone. And then once you've collected an entry, you can list all the entries that are on your particular handset, and then synchronize all of those data centrally. So this is a project we set up initially that's preloaded with the app that allows data to be collected, and this is a map of information that comes in. So this is a generic mapping interface that for any project, points are showed on a map, data are listed on the left-hand side, and then the results of the questions, should you have chosen to view charts, etc., are showed on the right-hand side. And this is just a time slider that allows information to be displayed based on when it was collected. So we've had about 14,000 projects have been set up on the first version of EpiCollect since it was released 5 years ago, varying very widely in scale or scope. Some of them include me demoing when it actually works, but here's an example of one that's been used for collecting information about vaccinations of dogs in India for rabies. So this was an NGO called Mission Rabies, and they attempted to vaccinate every dog within a month. So they created a whole series of projects. They captured dogs on the street, vaccinated, painted the nose, took a GPS position, and then synchronized the data. So they have maps showing where the vaccinations occurred in real time. This is a more recent example. The FAO provided training on EpiCollect for information about individuals who were or weren't suffering with Ebola. So they identified sentinel individuals within certain local areas, and they were tasked with checking a number of houses each day to make sure everybody was okay or not okay.
Where was that used?
Say again, sorry?
Where was that used?
Liberia. So this is a companion website that we launched. So that WGSA that I showed you for processing genome sequences, it's applicable not just to bacteria, but potentially to viral data, any other kind of information that has a genotype and can be tagged. So these were the data from Ebola. Again, we have a tree indicating the genomic similarity. And here we have, for example, the more recent outbreaks. Again, this is available for data to be added continuously. We really put this together just an exemplar of what could be done with the tools that we're developing. And then this is sort of where our plans are currently. So this is a genome sequencer. It's an Oxford nanopore. It's a MinION. It's that size. It's a USB sequencer. And we're working towards the combination of being able to sequence in the field, potentially. There are a lot of issues around DNA extraction and quality, but this is clearly going to improve linking to good metadata using mobile phone data, for example, and then linking back and forth with these kind of ways of placing a new genome in the context of all of the others that have been seen before, predicting resistance, etc. Two-way flow of information between mobile phones and web applications. I think I'm going to finish there. Just a lot of thanks to everyone in the Wellcome Trust. Any questions?
Great talk there. So, any questions, please? I did manage to tweet.
Oh, thank you very much. Hope it was didn't work. Demo.
No, not that. Yeah.
It's a lovely tool.
Thank you.
Just a simple question, just about the GPS component of mobile phones. You show that if you're doing things on a kind of national or international scale, you can identify where things are being done. But on a street by street level, is the resolution geographically good enough on a mobile phone? Or do you have to use
Yeah.
other
So.
type of GPS?
So I think it varies on the handset, but you can capture an accuracy value, which if you look on an iPhone, you have a kind of a halo outside, and you'll notice when you're hooking into GPS sometimes. That accuracy can be captured in meters, so it kind of gives a bit of a fuzziness. So you can keep recording GPS until you get a level of resolution that you're accurate with. I mean, it's down to meters is very, very reasonable.
How use of this in Liberia or in Sierra Leone, wherever the pathogen was, it's quite hard because largely the population only has 2G. So is this information downloadable for them or just for medics and researchers?
Oh no, it's focused on anyone with a mobile phone. We're not focusing or targeting medics. So essentially, the only time you need any data signal is when you load the project in initially. All of the data collection can be done offline. And then when you synchronize the data, you need mobile. So we've had, the FAO have also been using this across Kenya and Tanzania for resource management to identify facilities at various different locations. And they have phones with no contracts, no SIM cards, they just use wireless. So they'll load the projects in wirelessly, undertake data gathering, and then sync the data later wirelessly. It wouldn't really be a feasible application if it relied on having mobile data continuously.
Any more? If not, I'd like to thank you.
Thank you.